This lesson will focus on the Krebs cycle. Before you watch this one, hopefully you've already watched the Intro to Cellular Respiration and Glycolysis lesson, and you have a pretty good understanding of what goes on with those. Before you watch this, it's a good idea to have a look at those. Okay, so let's just quickly review. Recall, if you will, that cellular respiration is broken down into three parts: glycolysis, the Krebs cycle, and let's see if I can get my line, oxidative phosphorylation. Okay? We've already looked at glycolysis. Okay, and this is important. The products of each of these reactions kind of move on to other stages. Okay, so the NADHs, the two NADHs that were created during glycolysis, are now going to travel into the mitochondria. More specifically to the cristae of the mitochondria to help make more ATP through oxidative phosphorylation. Okay? The pyruvate that was generated during glycolysis is now going to move into the mitochondria as well, and it is going to be involved in the Krebs cycle to help generate a little bit more ATP, but more importantly, to help generate more electron carriers, NADH and FADH2, which are going to make the bulk of the ATP in oxidative phosphorylation. Okay? So, let's just quickly go over. Remember, what you want to take notes on here is the summary of the Krebs cycle. And you want to make sure you understand the reactants that are going in, and I'll point those out as we go through, and of course the products that are coming out, and any other important information that you think you should be writing down, do that, and then if you have any questions at the end, you bring them to class, and I will certainly answer those for you. So let's have a look then at the Krebs cycle. Okay, it's key to remember that the entire purpose of the Krebs cycle, believe it or not, really isn't to make ATP. It's to generate electrons that can be used in oxidative phosphorylation, which is going to generate a lot of ATP. Okay, so keep that in mind as we go through this. Okay, recall that glycolysis takes place in the cytoplasm. So we have our pyruvic acid created in the cytoplasm in glycolysis. It's going to go through a change from a 3-carbon compound. As you can see here, these blue balls represent carbons. Remember that glucose was a 6-carbon compound, pyruvic acid or pyruvate, remember those are interchangeable, are 3-carbon compounds. And don't forget, we produce 2. This is only 1. We'll talk about that in a second. It's going to go through a change into something called acetyl-CoA, which, as you can see here, is a 2-carbon compound. Okay, so we start off with pyruvic acid, which is a 3-carbon compound, and we move it to acetyl-CoA, which is a 2-carbon compound. In the process, we lose carbon dioxide. But we also make a NADH. Okay, and recall, NADHs are going to go off and be involved with oxidative phosphorylation. Okay, so before we even really get into the Krebs cycle, the pyruvic acid is converted or changed into acetyl-CoA. In the process of doing that, we lose CO2, and we create a NADH, which is going to go to oxidative phosphorylation to help make ATP. Let's talk about the cycle. Acetyl-CoA will enter the cycle. The very first thing that will happen is it will lose the CoA. Not important. Don't need to know, and again, don't really need to know any of these names, except for I will point this out. The Krebs cycle is also called the citric acid cycle because citric acid is, as you can see here, a part of the cycle. Okay, so Krebs cycle, Krebs is named after the scientist who kind of discovered it, and the citric acid cycle is kind of a more or I should say less vain way of mentioning this, because it's not named after a person, it's named after one of the things that's found within the cycle. Anyway, acetyl-CoA combines with this 4-carbon compound here to form a 6-carbon compound citric acid. You do not need to know the specific details of any of this, nor do you need to know the names of any of these. Okay? I'm just going through it to show you what happens, to hopefully allow it to make sense to you. But you don't need to be able to know the names of any of these things that we're going to go through. And notice that some of them aren't even written down here because they're not overly important for our intents and purposes. Okay. Citric acid is then converted into another compound. In that process, notice it's going from a 6-carbon compound to a 5-carbon compound right here. And in that process, we generate another CO2 molecule and some more NADH. Okay? That 5-carbon compound is produced down into, or reduced down into, a 6-carbon compound. And actually it's not reduced technically, chemically speaking, it's actually oxidized, because oxidation is giving up electrons, which is what's happening here, because look, the NAD plus here picks up those electrons that it loses and forms NADH. Okay, and again, another CO2 molecule is being given off. Several more changes occur. Notice, 4-carbon to 4-carbon, but in that process, an ATP is generated. Okay? An ATP is generated in that process. Another 4-carbon to 4-carbon, but a fat H2 is generated. Okay? And then a 4-carbon to another 4-carbon, where a NAD plus is turned into a NADH and a NADH is generated. Now, all of these NADHs, just like this one, are going to go off into, and a fat H2 actually, are going to go off into oxidative phosphorylation. Okay? The ATP that's been generated can actually go and do cellular work, because that's what it's for. Okay? Okay, now let's talk about so that's basically what's happening. Now let's talk about the whole thing. The sum, let's get the total for the Kreb cycle here, okay? Just erase this stuff for a second here. All right, so reactants. Remember, everything going in is a reactant. Everything coming out is essentially a product, okay, with these arrow things, what I mean by coming out and going in. Okay, so forget about this part, because it's technically not part of the Kreb cycle, okay, it's a precursor. Make sure you write notes about it, that this happens, this change happens, okay, but it's technically not part of the Kreb cycle, okay? So acetyl-CoA enters, citric acid goes from a 6-carbon compound to a 5-carbon compound, and in that process, a NAD plus, which is a reactant, okay, so acetyl-CoA is a reactant going into the Kreb cycle, NAD plus is a reactant, there's another NAD plus, there's another NAD plus, for a total of 3 NAD pluses. There's a FAD, one FAD. There's an ADP here for 1. Okay? So for 1 acetyl-CoA, 3 NAD plus 3 NAD pluses plus 1 ADP plus 1 FAD. Those are the reactants. Okay? Acetyl-CoA, 3 NAD plus, 1 ADP, and 1 FAD. Those are the reactants. Okay, I'm going to circle the products so you guys can see these. What comes out of this particular reaction or series of reactions? Okay, well, 3 NAD pluses go in, so guess what? 3 NADHs come out. And remember, NADHs carry those high energy electrons to oxidative phosphorylation. Okay, so we get 3 NADHs as a product. We get 1 fat H2 and 1 ATP. What else do we get though? Well, look at these guys here. We get 2 carbon dioxide molecules. Okay, so for the Kreb cycle alone, we generate 2 carbon dioxides, which is a waste product, 3 NADHs, and 1 fat H2, which are going to go to oxidative phosphorylation to help make ATP, and we get 1 ATP. That's per turn. Per turn. But remember, we actually created 2 pyruvates or pyruvic acids during glycolysis. So how many times does the Krebs cycle turn for 1 glucose molecule, which remember is what we started with? Well, the answer is the Krebs cycle turns twice. Okay, so every one of these numbers that you see here, okay, if we're referring to 1 glucose molecule, if we refer to 1 glucose molecule, everything in here, the Krebs cycle turns twice. So everything in here doubles. Okay, so for 1 acetyl-CoA, you get 3 NADHs, 1 ATP, 1 fat H2, and 2 CO2s. For 2 of them, you get 6 NADHs. You get 2 ATPs, you get 2 fat H2s, and you get 4 CO2s. Okay, so you're going to get questions. Some of them are going to ask per turn of the Krebs cycle, what are the products? Some of them are going to ask per glucose molecule, what are you going to get? You have to really pay attention to that, and you have to know in a little bit more detail what happens per turn as well as per glucose. Okay? And that in a nutshell, folks, is what you need to know about the Krebs cycle. Remember, though, the key, the most important part of this whole process is your generation of these electron carrier molecules, which are going to go on to oxidative phosphorylation. We're not trying to make ATP here, although we do get a couple, which is awesome. That's not the purpose of this cycle. It's to generate these electrons that are going to be used later on in oxidative phosphorylation. Okay, make sure you bring your questions to class, and we'll sort you out if you have any problems.